So we are representing biotechnology department, and the name of our project is cancer cell cycle. So as we know, cancer is the greatest one of the greatest urge of this industrial world, and cancer is most, it mainly divides uncontrollably, and we know the property of cancer, it is immortal. So here we have a basic cell cycle for a normal cell. So the first is G1 phase. In G1 phase, mostly the proteins, the enzymes or the nutrient which has been required for the cell has been synthesized. Next, it is S phase. In S phase, the main genetic material, which is DNA, has been duplicated and replicated. Next is G2 phase. In G2 phase, the main genetic material is condensed. Next is M phase, which is the main proliferative phase in which the two daughter nuclei has been formed. And if there are main checkpoints for this: G1 checkpoint, G2 checkpoint, and M checkpoint. It checks whether the DNA has been damaged. It checks whether the cell lacks the nutrient, and if the cells are mutated, they skip such type of checkpoint and further leads to cancer cell development. So let us take example of a UV radiation, which is one of the causative agent. Here we can see it is a normal cell. See, this normal cell has been affected with the carcinogenic agent, which is UV light. It is mutated now. As we can see, cell has the property of cell proliferation. It will further proliferate, and further it will give growth of a mass of a cell, which will further lead to the cancer cell development. So here, as I said, there is mutation in our DNA. There are mainly three genes involved in cancerous cell development. So first is proto-oncogene, second is oncogene, and third is tumor suppressor gene. First, proto-oncogene is the normal gene which is required for the development of a normal cell. Oncogene are the main tumor gene which is recognized for its cancer cell development. And tumor suppressor gene, as the name itself suggests, it suppresses the tumor formation. As we know, and we can say it is the guardian of our genome. So if there is any inactivation of this p53 gene, it will lead to the cancerous cell development. Here we have inactivation of p53. They are basically done by the three causative agents. First we have UVB. UVB is nothing but the UV radiation, which have a shorter wavelength, and it mainly affects the skin of our cell, of the human body. So it mainly leads to the cancer of the skin. So as we know, our skin is being underlined with the epidermal cell. It affects the squamous and basal cells and form a tumor. As you can see, there are patches here. These patches indicate the higher level of inactive p53 protein. And I would say the preventive measure in this is the one should use sunscreen and reduce the exposure to the UV light. Second, polycyclic aromatic hydrocarbon, which stands for PAH. These are nothing but the group of chemical which is being synthesized by fossil fuels. Fossil fuels are nothing but the coal, crude oil, coal, petroleum, gasoline. So if this polyaromatic hydrocarbon is being burned, it pollutes the environment, and the person who inhales it is mainly susceptible to this type of cancerous development, which leads to the lung cancer. And the person who are working in the oil refineries, which are like steel, iron refineries, coke refineries, they are more susceptible to it. And the person who are drinking smokes and alcohol, they are more susceptible to it. And the preventive measure for this, I would like to say that the people who are working in the refineries should wear the safety gears and the mask. Third is aflatoxin B1. As we know, the name itself suggests it is a toxin. It has been synthesized through Aspergillus flavius, and it is a toxic component. As we know, I would like to give an example of a agricultural product, which is peanut. As if this peanut is been produced through an pesticides or insecticides, if we inject this moldy part of a peanut, we will in turn lead to the liver cancer. It will directly affect to the DNA of our liver cells. So here, the main preventive measure I would like to say is we should use the commercial products, commercial plants, which has been made by the organic components, and we should consume it freshly. So at the end, I would like to say you should prescribe a doctor if you see any symptoms of the cancer. Thank you.
Good afternoon. Welcome to the PTC lab. I'm Shariva Kodu Patil. I'm from Department of Biotechnology, and today we have made our model based on the basic layout of a plant tissue culture lab. So plant tissue culture is one of our core subject, and this core subject's basic aim is to grow an entire plant from a small piece of plant, which is the tissue which we use. So the layout consists of 5 compartments. The first compartment is preparation room, where we prepare the media which is going to support the growth of our plant. The media consists of different chemicals like amino acids, vitamins, carbon source, nitrogen source, along with some inorganic salts. After the preparation and sterilization of the media, we enter into the inoculation room, which consists of a laminar air flow which provides aseptic conditions for the operator to perform the inoculation. In this laminar airflow, the sterilization takes place by UV lamp. After sterilization, a visible lamp is switched on along with HEPA filters, which blow sterile air towards the operator to avoid any microbes to enter into the hood. After inoculation, we take the culture bottles into the growth room, which provides all the optimum conditions for the growth of our plant. In the growth room, in the culture bottles, there is either formation of callus according to the media composition or different parts like shoots and roots. Now, if the callus is formed, then we further take it for cryopreservation if we want to preserve the callus, or for further liquid suspension of callus in which we grow the callus, and then after that we can extract any phytohormones, chemicals, or any pigments which we want to extract from the callus. After this, the cryopreservation room also has a hydroponics unit in which the water is in the bottom of the unit along with the nutrients, and on the top part of the unit there is a cocoa pit present in which we can either inoculate the callus or the seeds. After the growth of the seeds, we can either isolate the plants, mini plants which are grown, and then take them into the primary hardening unit. This unit is for acclimatization, that is, all the plants which we have grown are grown in the lab conditions which are optimum and aseptic. So the plant which we have grown is not yet exposed to the outside harsh environment, and if we directly bring it into the outer environment, it won't be able to sustain and will die. So to expose all the conditions which are present outside, like extreme heat, climatic changes, we bring it to the primary hardening unit, and over there we allow the plant to grow for a few weeks. After the plant is grown for a few weeks, it is ready to be taken out into the outside environment and to be planted by the farmers in the farm. Thank you.